Okay, so today we're diving deep into these biomolecular condensates, these tiny little droplets inside our cells.
Yeah, it's like a whole hidden world inside a world, isn't it? Almost magical.
It really is. We used to picture cells as having these rigid compartments, but condensates show us there's so much more going on.
Exactly. It's a whole other level of organization, a kind of fluidity that we're only beginning to understand.
It's mind-blowing. So these condensates, they're not like typical organelles, right?
Right, they're not static at all. They're constantly forming and dissolving.
Like tiny flash mobs of molecules, it seems.
Exactly. It's this process called liquid-to-liquid phase separation.
So it's kind of like, you know how oil and vinegar separate in salad dressing?
Yeah, precisely. But imagine it happening on a microscopic scale inside your cells.
Okay, I can picture that. So why is this separation so important? What's the purpose of these molecular get-togethers?
Well, think of it this way. If the cell is a bustling city, then these condensates are like pop-up markets.
Bringing together all the right ingredients.
Exactly. The right molecules at the right time, making everything super efficient.
Ah, so it's not just random mixing. Each condensate has a specific purpose.
Absolutely. They can speed up reactions, for instance, by concentrating enzymes and their targets.
So it's like having a dedicated workshop for a specific task.
Yeah, it's all about boosting productivity, just like a well-organized factory.
Incredible. No, it's not just about efficiency. It's also about organization. Like these condensates are creating order within the cell
Exactly. That's key for things like gene expression and signaling pathways, keeping everything running smoothly.
and making sure the cell can adapt, I imagine.
Precisely. Respond quickly to changes in its environment.
Okay, but how do these condensates know when to form and when to dissolve? Is there like a cellular conductor?
That's a question that keeps researchers busy, for
I
sure.
bet. It must be super complex.
It is. We're still teasing out all the signals involved, but things like pH, temperature, the concentration of specific molecules.
So it's not just a simple on-off switch.
Not at all. More like a whole symphony of signals.
Wow. And this dynamic nature, that's what makes condensates so fascinating, right?
Right. They can assemble and disassemble on demand, allowing the cell to adapt.
And this is where it gets really interesting because if we can understand this symphony, we could potentially manipulate it.
Absolutely. Imagine designing drugs that could target specific condensates.
Wow, the possibilities. Okay, let's talk about stress response. I read that condensates can help cells deal with stress.
Yeah, it's amazing how they can actually protect the cell from damage.
It's like they have their own tiny defense mechanisms, right?
Exactly. When a cell is under stress from heat, toxins, whatever, these condensates can sequester harmful molecules.
Like forming little protective bunkers.
Precisely, shielding vital components from the stress.
That's brilliant. You know, this whole idea of condensate forming and dissolving, it really challenges how we traditionally thought about the cell.
It does. It blurs the lines between what we used to think of as separate organelles and the cytoplasm.
Showing us a level of fluidity and dynamism we hadn't fully appreciated before.
Right. And it has huge implications for how we think about disease and treatment, too. It's like a whole new toolkit is opening up. Yeah, it's like a paradigm shift in cell biology. This new understanding is changing everything.
I can see that. But to be honest, I get a bit lost in the details. What are the key players in this condensate formation?
Well, you've got your proteins, of course. They're the building blocks.
Providing the structure.
Right, the framework. But the real surprise is how important RNA is.
RNA. I thought that was all about carrying genetic information from DNA.
To make proteins, right, that's the classic role.
Yeah.
But it turns out RNA is way more versatile than we thought. It can actually act as a scaffold.
A scaffold.
Yeah, bringing together multiple proteins, guiding the assembly of these condensates.
So it's like RNA is the architect, drawing up the blueprints.
Perfect analogy. And what's fascinating is that RNA can have very specific sequences,
Uh-huh.
and these sequences, they dictate which proteins it binds to.
Ensuring the right pieces come together.
Exactly. It's like a cellular matchmaking service.
With RNA playing Cupid.
I love it. And these matchmaking events, they're essential for fine-tuning all sorts of cellular processes.
Stress response, gene regulation, all that.
All of that. But then you start thinking, what if something goes wrong?
What if there's like a glitch in the RNA, a typo in the blueprint?
You're hitting the nail on the head because there's growing evidence that defects in RNA processing,
Oh,
they can lead to condensate dysfunction.
and that's bad news.
It can be, yeah. Researchers are finding links between these defects and a whole bunch of diseases.
Cancer, you said.
Cancer, neurodevelopmental disorders, the list goes on.
So RNA is clearly a VIP in the condensate world.
For sure, but there's also this specific type of RNA called long non-coding RNA.
LNC-RNA, right. I remember reading about that. They play a role here too.
They do. They're like the new kids on the block, and they're shaking things up.
New kids. What do you mean?
Well, unlike the usual messenger RNA, which carries the code for making proteins, these LNC RNAs,
Yeah.
they don't seem to directly code for anything. They're like transcripts without a clear purpose.
Mysterious?
Very much so. At least not a purpose we fully understand yet.
So what are they doing in these condensates? Just loitering?
Not at all. It's becoming clear that they have these important regulatory roles.
Influencing how condensates form and function.
Precisely.
Okay, but can you give me a specific example?
Sure, one that's been studied a lot is this LNCRNA called NEAT1,
Neat one. Okay.
and it forms a specific condensate called a perispeckle in the cell's nucleus.
Perispeckle. What in the world is that?
Think of it like an RNA prison, or maybe even a library.
Okay.
They sequester specific RNA molecules, preventing them from interacting with other cellular machinery.
Interesting.
And this can have huge effects on gene expression and how the cell behaves.
Wow, so these LNC-RNAs are adding a whole other layer of complexity.
They are. We're only just starting to understand their roles, but it's clear they're key players in the condensate game.
And all this has implications for both normal cell function and disease.
Absolutely.
As we go deeper into this, I keep wondering, what does this all mean for us?
That's the big question, isn't it? And the answer is, it impacts us in huge ways. This research is changing how we understand the cell itself.
Forcing us to rethink how we approach disease and treatment.
Exactly.
Yeah, it's like we've zoomed in from looking at the city to exploring what's happening inside each building.
Exactly, and that's what makes this research so exciting, uncovering the mechanisms within these condensates.
And to do that, researchers are using some pretty amazing tools, right?
Oh, absolutely. One of the most powerful tools we have is fluorescence microscopy.
Fluorescence microscopy.
You can think of it like attaching these tiny little glowing tags to molecules.
Like little beacons.
Yeah, so we can track their movements and interactions inside the cell.
So you can actually watch these condensates forming and dissolving in real time.
We can. It's amazing. We can see how different molecules come together, how the condensates change shape.
Incredible. So it's like watching this tiny, bustling city under a microscope. But how do you know if the molecules are just passing through or actually hanging out in the condensate?
Ah, that's where a technique called RAP comes in, fluorescence recovery after photobleaching.
Okay, I'm trying to picture that.
Imagine we tag a protein inside a condensate with a fluorescent marker. Then we zap a tiny area with a laser.
Tap it with a laser.
Yeah. Essentially, we make it go dark, then we just watch to see how quickly the fluorescence recovers.
Like if new molecules are moving into that spot.
Exactly. If it recovers quickly, it means molecules are constantly flowing in and out. The condensate is very dynamic.
Okay, and if it's slow,
Then it suggests the molecules are sticking around longer, forming a more stable structure.
so it's like measuring the foot traffic in our molecular city.
I like that.
High traffic, it's a bustling marketplace. Slow traffic, maybe it's a more exclusive club.
Yeah, great analogy.
So what other techniques are out there?
Well, there's super resolution microscopy, which lets us see even more detail.
Wow, even more.
Yeah, it's like upgrading from a standard definition TV to ultra high definition. We can see the condensate structure with amazing clarity.
That must be incredible to see. Is there any way to study these condensates outside of a living cell?
Yes, we can isolate condensates from cells and analyze their composition.
Like what molecules are present?
Exactly, and in what amounts. It's like taking an inventory of our molecular cities, seeing who's living in each building.
This all sounds incredibly interdisciplinary, pulling in techniques from biology, chemistry, even physics.
Absolutely. That's what makes it so exciting, and the pace of discovery is just phenomenal. Just a few years ago, condensates were this niche area of study. Now they're at the forefront of cell biology research.
It's like we've discovered a whole new continent in the cellular landscape.
And who knows what we'll find as we explore further.
Exactly. Well, I think we've reached the end of our deep dive into the world of biomolecular condensates. Thank you so much for taking us on this journey.
My pleasure.
And to everyone listening, we hope you've enjoyed learning about this incredible microscopic world.
A world where even the tiniest droplets can hold the secrets to health and disease.
Until next time, keep diving deep.